Hey there, thanks for joining us at the Rabbit Hole of Research, where you'll learn a few facts that you can impress your friends with at a party. Hey, welcome back to the Rabbit Hole of Research down here in the basement studio for the mini.
The mini.
Yeah. So here we are. You got me, Joe.
You got Nick.
We've got Nick.
Hi.
And we've got Georgia.
Wow. Great energy.
Very enthusiastic.
Hi. I'm here. I'm Georgia.
Didn't even say the name, as people know her. Just by the hi, like, who else is going to be?
You know this voice. It's Georgia.
You know this voice. Yeah, so last episode we talked about Splatterpunk, when Terra becomes entertainment. We had Freak was our guest, Splatterpunk author, and yeah, it was a good conversation.
It was, and you guys went to the fest, right, this weekend?
We did, yeah. So we went to Slay the Lake in Kenosha.
Slay the Lake, that's what it was.
Yep. LGBTQ plus horror book festival. So it was a really fun time taking a road trip to Wisconsin. In Kenosha, it was the final girl bar in Kenosha.
Oh,
it
nice.
was a horror-themed bar, so really cool. If you're in that area,
That's fun.
go check it out. It was really neat. Yeah, I had a good time. So met a lot of cool people, authors.
Very cool.
And while we were there, unrelated to fear and terror, was we went to the Milwaukee Zine Fest also. So we went there, and then on our way back, stopped in Kenosha. So a twofer.
Oh, very cool. Because, yeah, you guys were just there on Saturday, right? It was a one-day event?
Yep, Saturday. Yep, it was just a one-day event.
At least you guys were able to make the most of it. The episode itself, I thought it was a good one. It feels like it's in the Halloween spirit, but out of the Halloween time.
Halloween in the spring? I don't know.
Well, it's always
Summerween.
It's like...
It's always a good time for horror.
Yes, always a good time for horror.
There's never a bad time for murder. Yeah, I get it.
There is not.
Is that the message we're going with for this episode?
Phobias. Yeah, no, it was really good. A good episode. We had a listener comment, Alex. He suggested one of the body horror novels that he enjoys reading is Someone You Can Build a Nest In. The title sounded familiar, but yeah, I'm going to check that out.
That's interesting.
Oh,
so it seems like...
Who's that by, Dina?
It's by John Wiswell.
John Wiswell.
Yep.
And it's a newer book?
2024.
Okay.
Yeah, it's like a shape-shifting, weird fantasy romance novel.
Wow. That's got like everything.
This book is known for its dark humor, gruesomely wholesome tone, and exploration of themes like love, family, and identity from a monster's perspective.
That sounds interesting.
Looks like
I kind of remembered
won
Alex.
a Nebula Award for Best Novel and a Locus Award for Best First Novel. It was a Hugo Award finalist.
Wow.
Yeah.
Very cool.
I kind of remember Alex saying he doesn't watch horror.
I guess he reads
but,
it.
Yeah. And I was just talking to someone at my work, and she's really getting into horror books and reading horror. And then I asked about some movie, and she's like, "Oh no, I can't watch horror movies, but I can read."
Off mic, we're gonna have to talk about who this is, so I can tell them some good recommendations.
But like she was saying, like I can read horror, but I can't watch it, and I thought that was interesting. So yeah.
Yeah, that kind of goes. I mean, I think that maybe you can distance yourself in reading it. Maybe the visuals.
I feel like
really
seeing
put you
it
into
might be.
Right?
Yeah, that might be the problem. It's you can hear the gruesome stuff, but seeing it actually acted out by humans,
right?
But I think
might be where it's like, oof.
You begin to feel it a little more. It's like you see like leg injuries or arm, like you see people get hurt.
Some ways, though, I think your mind sometimes comes up with more gruesome than it feels. It feels it in even more so than what they might have came up with.
Yeah.
I can completely see that because then that's where you start seeing it reenacted in your head, and then it just stays there because you read it.
Mm-hmm.
Yeah.
Mm-hmm. Yeah. was going to say we had watched that Netflix series, Something Bad is Going to Happen.
Something very bad.
And so, was it very bad?
It had
was
a very bad. It had a very
Okay.
bad. Yeah.
I don't know. You guys have said it both ways to me.
I know, yeah. It's on Netflix. It's really good. It's 8 episodes, I believe.
Mhm.
But on the episode, the Splatterpunk episode that we did, the one movie that came up was The Monkey. And we hadn't seen the Monkey. George and I know we hadn't seen the Monkey, and we watched it. It was really good.
It
but
was really
something
good.
bad is going to happen.
Something very
bad
bad.
is going to happen. Had the same.
It really kind of had that
Yeah.
same.
Yep.
Yeah.
Vibe.
Yep. Kind of this generational horror passed
And
down.
kind of had to do with a curse and
Yeah. It was really good. Highly recommend both of those. The Monkey was really good. And something really bad is going to happen. Very bad. Which really bad did happen because I was in the cleaning the kitchen refrigerator.
And that was because you were joking
I
around.
was joking
and you
around
said
saying
that.
something really bad's going to happen. And then I shattered the,
Oh,
something really
fat,
bad
I went-
happened. So
So don't be just running around saying that willy nilly.
I mean, that's always the turning point in a story. And he's go, oh, I have a bad feeling about this.
Right.
Like, or I feel like something bad's going to happen. Yeah.
Right.
Something
right,
always does.
Right.
You're loading that gun.
yeah, never show a gun if you're not going to use it, right?
Exactly.
Coolio. All right. Yeah. I think that was it. It was a good, nice, tidy episode. Not a lot
Yeah.
to rehash. I mean, I guess we could, but probably that's a whole nother episode.
Yeah, exactly.
Not the mini. Well, cool.
Should we move on to new territory of studies? What are we
Yeah.
calling the segment? I can't
Uh,
remember. I thought we had a name for it.
Science segment.
Yes, that's
I thought
right.
the
We
whole
still
thing is
need-
a science segment.
No.
Come on,
This is
Georgia.
a science podcast.
No, but this is the specifically science.
Yeah, I don't know. I don't know. If we still have a
segment.
name for this. We just
Oh,
we'll
we gotta find
awkwardly transition into this segment. That's
Well,
it.
we got to come up with a name
I
for
thought we
it.
had something.
and maybe
What do we
like
have?
I
a
thought we
little-
did too.
And maybe like a little jingle that we can put in.
I know
right
that's
now.
what I
Oh.
put it out there for jingles. Like maybe I'll have to make something. I'll have to do it.
It'll go boom,
No,
boom,
that's-
No, you don't like my freestyle jazz, my mouth
Yeah.
jazz.
Let's. What do you got? What do you got? What have you been checking out? What's
I was reading something a couple days ago. It was a study where they were finding bacteria that fights off viruses using different ways. But all right, so I was reading this and then I don't know what happened, but I got really distracted. And I had the question for you. How small do things go? And do you know when it stops? Like how far, when is it the very furthest you can go?
I don't.
In
Well,
smallness.
small. What do you mean by small?
Yes.
Like
Well,
that.
Well, I
since-
mean,
Joe is the expert about
Yeah,
small
right.
things.
What's your question about size?
He's saying it's something like, is
Whoa.
it like go on infinitely small?
yeah. Like when do you know it's the very smallest it can go? Nothing else is going to be on top of that.
You mean living things or like
Yeah.
atoms and quarks and subatomic particles?
You're
like,
talking
what is our-
like Ant-Man?
What is like the quantum realm? Wait, well, I guess
Is
I
there
guess
a quantum realm?
what-
Yes.
yes, there is. Yeah, no, you can get pretty
there
tiny.
really?
Yes, there. I mean, yes, there are subatomic particles, so you can get down to the atom,
What's
and
the
then there's
smallest
things
that
inside
anyone's
of.
ever seen, like with a microscope?
I think folks have somewhat imaged electrons, and you can look at atoms, so you can see that. But yeah, I think I don't know, like if there's some quantum tunneling and things like that, some other techniques where you can image.
Like-
but what's the real question? Like, why do you know how small something?
That is
right.
a question.
But
what
I'm
are
saying,
you
like,
not?
you
That's not a
started
good
off
enough
like
question.
with bacteria and viruses and how small. So I'm trying to get to what is size?
I think
where
he's
are we
just
getting
saying
at?
that. I think that I don't know.
Well,
I should
because they're
like.
saying they found new things in it, even though they had used the same gene editing system to discover CRISPR and DNA snipping.
Sniping.
We've been sniping.
Sorry, I had to look that one up real quick, and I was like, wait, reread that. But yeah, it made me think, well, if you can find that already in what we see, can we go deeper and find better tools?
So I think what they did, those are all genetic tools. And so that's at the level of
Yeah.
DNA, which isn't at the level of the smallest particles in the universe. So
It's pretty small.
you're right there. I
right?
mean, it's subcellular,
It's cellular.
but DNA is a complex molecule. So it's not even individual atoms. And so what they're doing, and I don't know if this paper at all, but these systems like CRISPR, they were originally designed for the bacteria to cut up genetic material. So bacteriophages, so these are viruses that invade bacteria. You know, the bacteria need some defenses against these viruses that will infect them, the bacteriophages. And so they developed a system, the CRISPR system that we hear about in gene editing now. And it was originally used, and they use it to actually cut up the genetic material. So when a virus injects its genetic material to be used the cellular machinery to make copies of itself to make new bacteriophages, the bacteria goes, hold up, it has some defenses, uses CRISPR-Cas and then identify, destroy the invading viral genetic material. So that's how that system works as a bacterial defense mechanism against viral infections. And the idea there, and then after you cut up this DNA, this viral DNA, some of that can linger around and even be incorporated in the bacterial DNA to actually be activated as maybe like a genetic memory that it can then block new infections that comes along. So the bacteria has complex mechanisms to protect itself against viruses that might be trying to invade and destroy them.
But that's as far as we've made it so far. Yes, like we haven't gone deeper into the smaller structures, or no?
So I'm trying to
I know
reframe.
It's a really hard.
No, I'm trying to reframe what. Yeah, it's kind of because I think small is the wrong adjective.
Is it?
Yes.
Complex.
I'm trying to figure out the question, like, so the mechanism.
If
so this is
too
all
weird of a question. We can skip it.
Yeah,
it
no,
okay.
I guess we can try to hash through it. Yeah, because
these
these are
real bad. That
these are all gene mechanisms to these molecules that do work. So they're proteins, and they do work on things in the cell. So when you say small, it's not a matter of going smaller. You know what I mean? So maybe if I can reframe the question, or the idea is, are there other systems like the CRISPR system? Have they all been identified genetically? Right? There's probably bacteria using mechanisms that are to protect itself from bacteriophages that we might not have identified. So have we identified all of the systems that bacteria uses to destroy invading viruses? I don't know. And I would venture to say no. So I think we'll still find and mechanisms of how these systems work. And, that's why we do research, explore, and try to find that. So I think that's how I'm interpreting your question, more like, have we figured
that
out
actually goes closer to what I'm trying to,
because like you're saying, there's some new research saying, we just discovered this new system, but bacteria have been around for a long time and viruses have been around for a long time. And so
it's more a matter of we figured out a way to actually
understand the system
and manipulate it
for our own purposes. That's right. Using CRISPR. Yep.
But wasn't CRISPR discovered in a really unusual way? Like, and I can't remember. I just remember listening to a really good Radio Lab episode, but it was probably 10 years ago. And it felt like...
I don't even think it was 10 years ago. I think I know the one you're talking about.
And it was so good. I'll have to revisit that because it felt like...
I feel like that was recently, though. Like, within the past 5 years.
I think it was longer.
Yeah, I mean, CRISPR was, I think if you timeline it, probably the late 80s, early 90s. So
really use it and stuff.
Really use it, right. I think that was mid 2000s. So yeah, so you're probably right.
I know that it was a few years before the pandemic.
Yeah.
That I remember listening to that.
I think that's when it really hit. And
and seemed like there was some really interesting, like, that it does have a very interesting scientific history, you know.
Yeah,
That whole-
I think it was just discovered in bacteria as part of their immune response, and these clustered repeated sequences, and they didn't know why they were repeating and the purpose of this genetic repeats. So I think that was it. And I think it was a little bit later until people figured out how this could be acquired immune, kind of we were just talking about earlier, that is taken in these snippets of viral genetic material and then using that as a memory, genetic memory, to then when you see that pattern, the viral genetic material comes in, it sees the viral DNA RNA pattern in that, and then it uses this protein. CRISPR cut that up, right? So you're matching the pattern and then cutting it. And then what we use this system for gene editing is that you can then go through, find a pattern, and then cut out and replace with a different piece of DNA.
So we're basically using something the body already does.
Bacteria.
And then, something that bacteria already does.
This is a bacterial system. CRISPR is a bacterial system.
Within our body.
Right, because DNA is DNA. The molecular structure of DNA, the same nucleotides as they're called, are used in bacteria and viral, fungi, plant, like every life form on Earth that we know of uses DNA.
Oh, I like
and
the way
it
you
has
said
the same
that we
structure,
know
so-
of.
Right. I mean, I'm not gonna paint myself in a corner. There's always something. But what's the odds of that? I don't know. I'm not going as far as the universe either, right? Because we haven't done that.
Not yet.
But that's kind of how this works. And we have in our own bodies, we do have mechanisms to cut and remove DNA, the same thing.
But to be able to realize that and then use it in these clinical...
Yeah, it took
like
decades of work. I mean, like I said, I think it was late 80s when they first realized this pattern, but didn't understand what the purpose was. And then you had all this research to then utilize this and then make it a tool and now push it. Like, how far can you push this tool to not only to remove DNA and go and edit and replace DNA, but to epigenetic kind of changes. So epigenetic, remember, are these kind of overlays. So you have your genetic information, and then you can have other molecules that will turn things on and off, called methylation. So you add specific markers that say, okay, turn this one on, turn it off. And those epigenetic kind of traits can be passed on. And now you can use CRISPR to actually modify epigenetic kind of markers. So can you turn genes on and off like that? So not replacing a gene, but kind of going in and say, okay, let's turn these on and off and then get some
and just,
change.
Doing slight changes where it's not going to be
right.
something crazy.
Yeah. So I think it's really cool.
I think the whole idea of CRISPR is really neat. Like every time we talk about it,
it is fascinating.
Yeah.
It's something that sounds like it's science fiction.
Yeah. No, I think that's in that book. Change Agent that was CRISPR. Gene editing was a big thing. And then especially epigenetic changes, because you think mostly of the changes happening embryonically when you have fewer copies of the gene, you're early on, you change it, and then the cells as they replicate will have the new quote unquote healthy copy of the gene you're trying to edit and replace. Epigenetic in an adult, can you modify gene expression and
is this
regulation?
This is probably a dumb question, but is that related to certain like illnesses? Like certain, so is that similar to being predisposed to something?
So like epigenetics has really like environmental stresses can cause. So really what you're doing is you're modulating gene regulation. So you're turning on and off or how much to produce. You're actually turning on and off a product, a protein, and that protein is doing some work in the cell. So
and
if
that
you're
could lead to some
that's right.
sort
It
of...
could lead to disease, stress, anxiety, neurological disorders. So if you can now go in, understand what's being epigenetically controlled, can you go in now and turn off, turn on, something that should be off.
Really weird. So. Michelle had said that word earlier, and I did not know what it meant until 40 minutes ago?
Epigenetic?
Yeah.
Yeah. We've mentioned it on the podcast quite a bit, so just need to.
I feel like we do, but I never grasped.
We just don't listen to me.
Right.
Yeah, no, it's really interesting.
I'll leave you. I'm not Georgia.
It just takes a few times for it to sink in, right?
Yeah.
Yeah, she actually gave the definition, and it was like the only thing I was learning at that moment.
Yeah. Yeah, and I think we touched on this, swing all the way back to bacteriophage and why kind of the interest in it and how bacteria use their systems to fight that. Because as we overuse antibiotics, you might need alternate therapy to fight bacteria. So can you use bacteriophages to actually attack and kill bacterial infections that you may have? This research is leading to something beyond even this CRISPR.
Sorry, I think I threw us off.
Yeah, no, yeah. You said small, like you were. I think you were. You know, it's not a matter of size because that wasn't the key. So sorry, we had to unpack. Terminology there, which is okay. You know, so
words are
then
hard.
because then we got into like quirks and stuff, and that's a different area of science. But I think you wanted to be more in the biological cellular function.
Very cool. Did you have one, Joe? I'm sorry again.
No, yeah, I had a couple that I saw that were interesting, and I think some because the title was like interesting, and then I poked around, but one was harnessing semen-derived exosomes for non-invasive drug delivery. And so, yeah, a little bit to unpack. So exosomes are these kind of very small vesicles. So vesicles are like these kind of lipid droplets that the cells excrete, and they can have information. They can have genetic information, protein information, and that transports between your cells. And they use this kind of as a way to pass notes, some DMs. So they're just passing these little messages between each other, these kind of exosomes. So they're exo, outside, some cellular or body. So every type of cells make them. And so what's interesting is that in this particular study, they're using this to make kind of these eye drops for a tumor growth in the retina, and in particular retinoblastoma, this rare malignant eye cancer, really, really prevalent in children. And so how do you treat this kind of eye cancer? And so the way they do it now is chemotherapy, injections, radiotherapy, all these pretty invasive procedures because you have to get in through many different layers of the eye to get to the cancer. So what these researchers hypothesized and tested was if you take exosomes from tissues that have to pass through many different signals through different tissue types. So in this case, the semen used for fertilization. So it's got to go to a different system through different female reproductive tissues to reach its target and fertilize and do reproduction. So they said, well, can we use these exosomes that can have these abilities? Do they have any special properties that allow them to pass through different cell types and different tissues?
Absolutely love to see the people behind this talking about it.
Yeah.
Like,
so
just to come up with this idea, they had to say some weird shit before they got to this, Joe.
So yeah, so they do that, all the research to get there. That yes, they do have special properties that will allow them to penetrate and move through, just like they do the female reproductive tract. And so can you use this in the eye in kind of the same system? Can you kind of, once again, like the Cas system, the CRISPR system we were talking about, can you take a system that has another function and then repurpose it for some other biological purpose that you need? And yeah, it seemed to work. That I believe this was done in mice, where they tested it, that they could actually take these kind of...
It's kind of like a superhero.
Semen-derived exosomes, SEVs, and...
What was that,
through.
Georgia?
No, don't you picture them like little superheroes that can
little
like
exosome superheroes?
Yeah, they can like bust through different structures and get to. It's like
I don't think they're busting through. I think they're just passing through. Yes,
yeah,
right.
I still think they're like little.
It's not semen. It's just
like
little vesicles, so they're isolated.
Like little superhero vesicles.
It's no weird. Yeah, these are isolated, so they take it. They're purified. These are what the semen cells are excreting, the exosomes. So then you collect that, and you're not putting sperm in someone's eye. So let's clear this all up, Nick. So I see you giggling in the back of the class. Yeah, it's a really cool study. Yeah, that they have. So it's really promising because you can deliver now. Can you take these exosomes, load them up with anti-cancer treatments, and then deliver them without kind of these kind of very invasive techniques? So can you do this and have high efficacy? So
I do agree with Nick. It is fascinating how not just scientists, but anyone working on a problem, solving a problem, and to get to the point, you know what I mean, where they were able to think about this. That's like, that is fascinating.
And a lot of research has went into that. So from these other systems, like how do exosomes work in their natural environment? How do they work there? And then go, can this...
And I can just see somebody like waking up like first thing in the morning, like, oh my gosh, I got an idea, like, I don't know. It's really...
Yes, I don't know. I'll put the show notes. I'll put
also,
everything in show notes.
It's similar, like you said, it's similar to like architects or engineers looking at like things that happen in nature and then saying, oh, that's a great system. Let's try that could like solve whatever.
Try to emulate it.
Right. Yeah, definitely. Yeah.
So then this is taking that same idea, but on this
at
much
this cellular level.
Yeah. It's very,
You were going to say small level.
Well, no, I was not.
I'm sorry.
No, you're fine.
It is small.
They are small, right? That's why, so that's why where we headed with the smallness. But yes,
Small
I think
things are small.
Yes, they're small things. Yeah, another one I thought was interesting, and once again, it's answers blowing in the wind, detection of birds, mammals, amphibians, and with airborne environmental DNA. And so they did this survey, this group. And this was a couple years ago, and it's just coming back up again. And the idea is that everything you do in every organism is leaving some genetic information. It's just you scratch your head or your skin cells. It's just out there in the world. And so this team, they went and they tried to collect this airborne dust and then say, well, can we tell what organisms were in there, like what animals, what plants, what things pass by this patch of ground, and kind of catalog that. Like, is it sensitive enough? And so it was really cool because they could do that. But it raises other questions, like, and they point this out in the paper because you see some, I think one of the study areas, there's been several groups doing this, but one of the groups did this and they found salmon DNA some miles away from where the water source is at. And then they just realized this was carried, so right. Or how did it get there? So, because the salmon didn't walk
something ate
through the
salmon,
forest. It could have happened. It could have just blown through the wind. It could have been rain, all sorts of things that could have passed this DNA around. So when you do these studies and you do these surveys, you do have to be conscious of that. If you find traces of rat in some area, was that rat there? Or did it come in the feed? So did it pass a year ago? Like, you don't know. There's no time stamp on this. So how long ago did this thing pass? Like,
right. Like you wonder how long does this stay around? It's
really cool. And
but what's the practical?
So you're doing surveying, like, so if you're looking for invasive species, so is this organism invading in this area? We don't see it now, but was it here? Is it moving? You could think about it. They did this a lot with COVID. And they started looking at municipal wastewater, and then you can look for spikes. So you do a study of the wastewater, and you can see what genetic material's in there. And if you see a lot of COVID genetic material, then you're going to have a spike potentially in this area because now you're seeing more wastewater that has viral kind of DNA in it. So you can use it for population surveys to get some idea of what's happening in an area. But as naturalists, like I said, you can be out looking for species. Is there an endangered species? Do we see any trace of it? Has it been here? What's its movement?
It just really does seem like looking for a needle in the haystack.
I think using this technique is saying that you now have a way to find the needle. Like before, if you go, so how else you set up cameras and you look, and if the creature happens to go by, we see one. I think here you can now go out and do the survey and go, okay, what do we see? Oh, look at this. We do see DNA of whoever X, Y, and Z. So I think that's how you can think about it. Something has to tell
you to look in this specific area in the first place.
You could just be surveying, right? That's the nice thing of it. You can just survey even more areas. So, right, you set the cameras up in the wrong place, you never catch it. So here, can you survey and then know where to actually look for the thing you're looking for? So how do we make the haystack smaller? I mean, you can think of it that way. So I think there's a lot of
searching for certain, like maybe missing people or a forensic kind of application.
Yeah, I guess. I mean, like biological weapons might be one where you can use it and get a handle on it. But yeah, yes, you could. I think people are already doing DNA stuff like in the crime scene where people are missing.
Well, right.
Because you would need to know
course.
where they're going. I mean, that's even more like to your point, like if you're just sourcing down the street, I mean, if you happen to have
what
been
are the
there.
chances that?
Yeah, or false positives, right? So if they followed you in your coffee shop, workplace, train, these places you go a lot, then if you're taken from one of those places, then it really now, how do we find a new trail? So if you're looking for a person, I think. But yeah, I guess you could. Like if it was like, we think you're in the field, maybe they can do sources or something. Yeah, interesting.
Yeah, I mean, I could see it having other uses.
Yeah, I think it's more like this paper is mostly like monitoring species level kind of thing.
Yeah.
So not individuals in the
I know.
population.
It was kind of a far reach, but
Yeah. Oh, maybe like a science fiction novel or something. Cool.
I think that would be fun.
Yeah, we're right.
So what kind of media have you guys been consuming?
Oh, go ahead, George.
Oh no. What kind of media? Yeah. I'm sorry. I'm drawing a blank on anything.
Well, we just finished something very bad, really bad.
Oh, well,
Horribly
we already
bad
talked
is
about
going
that.
to
We
so
already talked about that.
Yeah, I kind of watched that. You should say you know what Georgia watched. She hadn't watched it.
Yeah.
And I was like, okay, we just got to watch it. You go ahead.
Oh, yeah. I watched Blair Witch.
Yeah.
Oh, really? You've never seen that?
I had never seen that.
What did you think?
No, I thought it was really good. I think I had so many preconceived notions about it that I thought it was very interesting what it really was. You know what I mean?
Wait, what were your notions about it beforehand? I need to know now.
Well, I mean,
Just so you had them.
I mean, I guess I won't be spoiling it.
No,
It's pretty old.
it was 99. So.
I'm feeling so old right now. Oh my God. I think I thought that it was going to show more like we were talking about, like maybe show more gruesome things and then it's like the whole movie I was waiting for kind of these jump scares or these like more gruesome
Oh no.
And
it's
so
more atmospheric.
it is definitely more what you make it in your mind.
Yep.
Exactly.
Which I thought was really good.
Yeah.
So I was surprised that is what it was.
And it holds up really well. I hadn't seen it in a while.
Right.
I saw it in the theater when it came out. Some of my neighbors, they were going to see it. And I had no clue what Blair Witch was about. And I went in and it was really, it was good. And then watching it now, because it's been a while since I've actually seen it. So putting eyes on it again and going through it, it was like, oh, this really holds. It was done well. And I think the simplicity of it, that you aren't relying on kind of gadgets and graphics and practical effects. It's more just these people, they're just trust. They're kind of.
I think that does hold up. But in a way, it's like we're so used to like things getting more like what we talked about in the episode, how now it's almost like you really have to make something like extremely gruesome or extremely out there because you're trying to like.
I don't know if you,
and then I think that's a good reminder that you don't have to.
You don't have to. I was going to say, I think there's something to be said with skillful writing, acting, putting people in these positions, and then having them limit the information that the viewer has, and then using that to drive tension and suspense and the horror of it all, and putting yourself in that situation. If you're in the woods with your map that's not right, your friends, their association was loose, so they weren't like best buddies or a couple or some serious partnership. They were just going out there to do this project and help a friend out. And now you're stuck in a situation that how easily these things fray and come apart. And yeah, it was cool to watch. And if you haven't seen Blair Witch Project, go check it out. The first one in 99, or if you
honestly,
have seen it,
the third one is as well.
Is it
It's
good?
actually,
I haven't seen any other ones.
The third one is really good.
Yes, it's not labeled the third one or anything like that. But because after the original came out, Season of the Witch,
right?
And that one, I don't remember
Yeah, I didn't see that
So
one.
I'm assuming it wasn't great. But the one that they did in 2016,
Mm-hmm.
And
that was pretty
then
good, where they brought it to a more current day setting. So they had technology and that was failing on them. And it was
Right.
really
Oh, I
cool how
see.
they were able to
Mm-hmm.
still bring that
I
feeling of alone
I
and
think they
stranded.
I think they called that Blair Witch 2, right?
No, it was just called Blair Witch.
It was just a
Oh.
season of The Witcher or
Because
something like that. Yeah.
We noticed the
Maybe
other
there was
ones.
a 2 or something.
There
Yeah,
was
I don't know. Yeah.
one of them said like had the
It was
number
The
2.
Blair Witch is the first one.
Right.
Yeah. I
The
don't
Blair
know.
Witch, yeah.
Mm-hmm.
And then the second one was, Book of Shadows, Blair Witch 2.
Okay.
That's what it was.
Yeah.
Okay.
And then just Blair Witch. Yeah.
It's funny because that kind of gets back to that whole thing about watching a horror movie as opposed to reading a horror book. And that kind of gets back to that whole thing of how much do you show?
Yep. Mm-hmm.
And then I think of those movies, I can't even think of the name of them. Is it Mist? Or those movies where there's some sort of creature out there, but then
did that. Yeah.
and then they show this
Yeah.
real cheesy thing,
Yeah,
and then
yeah,
you're like, oh, well, that's it. You know,
They had
it's
to
just
save
like,
on the special effects
and
budget.
it's like, no, just don't show the
right,
thing.
Right.
You know what I mean? And then that
Let
makes
the mind kind of,
fill in
Yeah,
those. Yeah.
And Blair Witch did such a fantastic job with that. Like, I remember seeing that as a child on VHS,
Oh,
where
wow.
it still
Yeah.
sits with you because you're like, oh,
Mhm.
you don't know what this creature is or
Right,
what's going on.
Right.
Yeah.
Right.
And then they also had a video game too.
Interesting.
That was pretty good.
Oh, wow.
The other thing
I
we
think
did
I own that
at the
on multiple systems.
Oh, go ahead. At the Slada Lake, at the Final Girl bar, we were sitting there eating a little food and chatting with a friend who happened to come down for the festival. And they had Friday the 13th, Part 8, Jason Takes Manhattan. And that's one of my favorite scenes. And we started watching close to that scene where you have the black guy. He's in this like track suit and he like starts boxing with Jason on the roof. And they go, and then he's winded, he's gassed, and he's like, take your best shot. And then Jason punches his head off. Like, it's like one of the, it's just that over the top. And I think that's,
Oh my God. And like we were talking about that movie isn't even a horror movie. Really.
I mean, it's
not
kind of fun,
scary.
right?
It's just silly.
I think it was
It's
like you got
just-
into
you
like
get in
how,
that whole range.
how can you kill people? Like, what's the funniest and most unique way? What haven't we done yet? I think that. And Freddy Krueger did the same thing when you got into the 4 or 5. It was just, how do we kill somebody with a spool of Scotch tape or something? You know, it's just being silly.
You have to up the antes because it's just, yeah, and that's what it just becomes, which I like it.
It's a comedy.
Yeah, it was fun.
point.
I hadn't watched that in a while. Like, I knew it, and it was like, oh, I think that scene's coming.
And we were debating the year it came out based
That's
on
right,
how high the hair
was. That's right. It was
and,
like,
but me and Glenn were totally right on about the about the 1980.
I said late 80s. Yeah,
came out 1988 or 89.
Right, yeah. I said early 90s because
No.
the hair wasn't as high.
No,
I
you
did say that. I said early 90s because I
You
thought the
said
hair
90s.
wasn't. I said early 90s because the hair, it was 88.
I don't know. This one feels like something
That's,
that Joe didn't say, but he's saying he said.
Yeah, they know I was right. No,
Yeah.
So what are you watching? Anything?
What I have been watching the new SNL UK. Been enjoying that a lot. I think they just had episode 4 is what came out. I think the crew is doing a great job. Yeah, definitely recommend that.
And what do you watch
else?
that on?
We've been watching that on Peacock.
Okay.
Mm-hmm.
Yeah, it's nothing too much to do, but it's been an enjoyable show so far.
Nice.
And then we have also just refinished 9.
I've never seen that.
That was a good show. I mean, it's a show about cops, but it's one that is a comedy show with Andy Samberg. So if you are a fan of him, they do a good job. And then I have just jumped back into playing the game Phasmophobia. ghost hunting game, where they had announced that they are going to be doing a crossover event with another really good game that I like, Alan Wake 2. So, yeah, just been trying to jump back into that whole series.
Cool. That sounds really fun.
Yeah.
Mm-hmm.
And then, just trying to get my readings done. Georgia had recommended a book to me that I just started.
Oh, you have
And
started it?
I just started it, yeah.
Okay. Yeah, we'll talk about that later. I mean, I think I'm
What's
like,
the name of the book again, Georgia?
it's Strange Animals. But
And what did you think of it?
oh my gosh, I'm loving
This
it.
is-
I'm
This is
loving
also a
it.
book that Georgia recommended to me, so
Yeah. So does that make it, is that good or
I
bad?
we're going to find out now, aren't we?
Yeah.
The pressure.
I mean, I recommended a book to you before, so you know.
Mm-hmm.
Yeah. And then I'm about 70% done, and I really like it a lot.
Oh, I thought you finished it already.
No,
Are you waiting for
because-
me to finish it?
No, when I recommended it to you, I was only like on the 5th chapter, so it was kind of a risky recommendation. But I was that
I know, look at
confident in it. It was that good. It was like, and I was that confident that you would like it that I recommended it that early on.
Man,
So yeah,
it's a really risky one right there.
I know. I think it's going to pay off. But
Good. But yeah,
Cool.
that's where I'm at.
Yeah. So I was going to just mention, maybe people have seen us online, but I was recently named one of the 35 writers to watch by the Literary Guild Complex. And they're having a celebration event April 30th at the Epiphany Center for the Arts. So if
and it
folks
is
are-
kind of a fundraiser for
fundraiser.
the
Yep, and you'll meet all the other writers that are there and other folks in the area, I guess, that are going to come. So yeah, this
And
is
all
gonna
of
be a
them are
fun
under
event.
35, right?
That is not the requirement of the
No?
list, but
35 under 35?
just 35 writers to watch. So they're going to be doing it. So yeah,
Yeah,
no,
it's
it was
35.
kind of a surprise and honored. So yeah, I'm looking forward to the event. But don't I throw it out there? Folks are looking for something
buddy,
to
you
do
deserve
April
that.
30th, and they can come on down and say hi, to have a toast, and celebrate all these wonderful. I've been looking them up and just really honored to be named among them. So yeah, throw it
Well,
out there.
as I said again, Joe, you deserve this one.
Well, thank you.
Not all your awards you've deserved, but
Yeah,
this one you
well.
have.
So I give myself.
Oh, I'm going to give myself this Rabbit Hole Research Award.
Yes, that's right.
I think the
I
only
give
person
awards out.
I
don't-
was going to say,
yeah.
I think the only person that's gotten a Rabbit Hole Research Award is Jeff Goldblum.
Jeff Goldblum. That's right.
That-
I thought we agreed it was Sigourney Weaver, no?
well,
It was Jeff Goldblum. We hashed this out.
yeah.
It-
In the end, Jeff Goldblum was
We had a word, and then we
the
gave it.
debut award.
Yes, and then we had a word of the season last season.
Well, that's a given.
Yep. So
What was the word for the season? I can't remember. I think it was what?
Shut up. You remember.
we'll put it in the show notes.
Extremophiles. But I kind of like this new word. What is it? I don't remember. But I still see
I don't think it'd
Exosomes?
be a new
exosomes. And they're like little superheroes. Exosomes.
Exosomes. Yes.
I just
Okay.
see them like with a little teeny tiny cape.
A little fist extending out,
Right.
passing through.
with a tail flying behind it, all slimy and whatnots.
Yes. Okay. Well, you've got me, Joe.
Yeah, I got Nick.
Got Nick.
George.
Got George. A little more enthusiasm at the end.
And we went down some mini hole.
Went down the mini. Stay safe. Stay curious.